Dr. Browder, should we leave these in there still? I just out of that. I don't know why.
Yeah, that's okay. Normally I would have had you pull your blood auger plates out of the incubator because they've been in there a while, but the blood was contaminated. I made a second set of plates and they had growth too, and I did a control where I did just the auger. So we've ordered new blood. We'll kind of have to play it by ear in terms of whether you'll be able to use those plates on that project for your skin and nose and things like that. But we can't do anything until we get back from break. So those plates are going to be pretty much useless. You can tell a couple things by looking at them. The growth that was contaminating them, none of that was hemolytic. And some of you, if you look at your plates, you're getting areas of hemolysis. But what I'd like to do is restreak those plates from some material when we get back. So don't worry about those. Tomorrow in lab, we're going to be doing an exercise on counting microorganisms. You're going to be doing a standard plate count. So that's exercise number 11. Probably won't take the full period of time. You'll have to come back later after break and do some counting of your plates. So if you do need some extra time to kind of get caught up on anything regarding your unknown or your peer culture or your throat, nose stuff, skin, there should be some time on Wednesday to do that. I will have your exams graded by noon and have them turned in as part of the midterm grade. And so your midterm grade basically will be your first two exams. That's not anywhere near half the points in the course. And so this midterm grade really isn't the midterm grade. You'll have quite a few points you'll pick up right after break with the lab exam coming up. Your first turn in of pictures of your known organisms. That'll get you some easy points if you've done that. So midterm grades are a little bit kind of irrelevant in this course in terms of where you stand midterm. So questions? Okay, for the rest of the semester now, we're going to get into topics that probably are the major reason why you took a course like this. Most of you have interest in some area of medical application. Not everyone, but a lot of you. And so one of the things we're going to spend the rest of the semester talking about is the ability of microorganisms to cause disease and the body's response to those disease-causing organisms. So we'll talk about infection and immunity. At the beginning of the semester, you read an article, and the bottom line in that article was that our relationship with microorganisms is much more complicated than we had ever understood prior to about the last decade. Part of that understanding is a result of a huge initiative called the Human Microbiome Project, kind of the microbiology equivalent of the Human Genome Project. And the goal of the Human Microbiome Project is to try to catalog the types of microorganisms that occur in various microhabitats of the body. And that's one of the things we will talk about, is that the body is not just one uniform habitat. It is not a 37-degree C incubator where conditions are exactly the same all throughout that incubator. You've got all these little places in your body where conditions vary, and consequently different microorganisms will inhabit those different parts of the body. And cataloging all of those microorganisms associated with varying parts of the body is really challenging. And it's really only been made possible by advances in advanced sequencing, which really has been made possible by high-speed computing and databases. We don't identify organisms much anymore by just culturing them on agar plates and identifying them the way you're trying to do your unknown. Now it's all about genetics and sequencing. And so consequently what we're doing is we're looking for novel genetic sequences as an indicator of novel species. And as we catalog more sequences, we can assign names to those particular species. Now, to make sense of all of this complexity of association of microorganisms and humans together, it sometimes helps to put a little bit of structure to trying to define those particular relationships. And so that's what this particular slide is trying to lay out for you is, what are some of the common types of relationships that we find between microorganisms and hosts? And one of the first big dichotomies in relationships is the relationship symbiotic or non-symbiotic? Okay? In non-symbiotic relationships, the organisms are free living, they are independent of one another, and the relationship isn't required for survival. But that doesn't mean that the relationship between species A and species B isn't important. Two types of relationships that we'll make reference to in non-symbiotic microbial associations are synergism and antagonism. In antagonism, one member can inhibit or destroy another. In synergism, members can actually cooperate with one another and kind of reinforce one another. We'll talk about antagonism when we talk about our body's normal flora in a little bit, and the role that our normal group of microorganisms plays in fending off some of the organisms that might otherwise be harmful. So these are relationships in our context primarily between one bacterial species and another. Okay? So these non-symbiotic relationships. We're also going to be very interested in symbiotic relationships. That term literally means living together. Bio-life, sim together. Okay? These are organisms that live in close relationships with one another, and the relationship is usually nutritional. One member of the pair provides nutrients for the other. In symbiotic relationships, the relationship is a requirement for one or both members. The types of symbiotic relationships we're going to be interested in are the human host, us, and microorganisms, especially potential pathogens. So if we look at symbiotic relationships, there are three major categories of symbiotic relationships: mutualism, commensalism, and parasitism. And we're going to look at these relationships in the most simple context. Two things interacting with one another. And we're going to represent the outcome of that interaction by the symbols plus, minus, and zero. So plus will indicate a beneficial relationship to one or other of the organisms. A zero will represent no effect, and a minus will obviously represent a negative effect. Okay? So how would we represent mutualism? Well, mutualism is a dependent process where both members of the association benefit from the relationship. So that would be a plus designation for mutualism. Now, we've already talked about mutualism in the context of nitrogen fixation, and particularly the relationship between legumes with their nodules on their roots and the nitrogen-fixing bacteria that can inhabit those roots. Both benefit. Okay? Plants get nitrogen in a form that they can use, ammonium. Bacteria get carbohydrate from photosynthesis. They're interdependent upon one another. Do we have mutualistic relationships between ourselves and microorganisms? Absolutely. And that is very easily seen in some of your gut or enteric bacteria. Those enteric bacteria get a free supply of pre-digested food. It's a pretty good deal, okay? We ingest complex molecules, and then we have a digestive system that breaks them down into things that can enter the bacterial cells and support their metabolism. So they can get sugars, they can get amino acids, they can get lipids. What do we get? Well, some species of bacteria produce vitamins, for example. Vitamin K is an important vitamin that we either have to get through our diet by eating plant or animal material that contains vitamin K, or this symbiotic relationship that involves microorganisms producing that particular nutrient.
This might be a dumb question, but at what point does a child get these bacteria? Because I mean, you're not born with bacteria.
Absolutely, and that's something we'll talk about in just a little bit. How do you acquire these bacteria that are beneficial? Very early on. But it's interesting because your normal flora undergoes a real evolution and its composition changes and matures as you age. So it's not the same. I mean, you begin acquiring it basically right as soon as you're born. But it's not going to be the same as when you are 5 years old or your age right now. It really goes through an evolutionary process of new species replacing one another. If any of you have had ecology, the process of succession where one species of plant particularly replaces another, your microbial flora undergoes a successional process where one group of species replaces another as conditions change. So it's pretty interesting. Okay, commensalism. What's commensalism? That would be a +0 relationship. Okay, one benefits, the other is not harmed. A very good example of a commensalistic relationship is staph epidermidis growing on your skin. Staph epidermidis can live off of nutrients provided by dead skin cells which are being sloughed off. You're constantly losing skin cells. That's what most of dust is skin cells. Okay? Those organisms obtain nutrients from those dead skin cells, and so they benefit. But is it any consequence to you to have those bacteria living on your skin in that commensalistic relationship? No. Okay? You don't even know they're there. And you would probably never know that they were there until you did the skin swab and streaked your mannitol salt agar plates, and most of you probably got staph epidermidis growing on those particular plates. They're the non-mannitol fermenters. Parasitism, that would be a +- relationship. The parasite obviously benefits. How? Nutritionally. They obtain nutrients from the host. The host may be damaged as those parasites grow. And if the parasite causes damage to the host, then we refer to that parasite as a pathogen. Now, are all parasites pathogens? No. Some parasites can establish kind of a détente with the host, where they're obtaining some nutrition from it, and so kind of technically doing a little bit of damage, but not much at all, and so little damage that the host isn't really even affected. If an organism penetrates the tissues and multiplies within those tissues, then we're said to have a state of infection. The organism that is causing the infection is the parasite. But does infection always result in disease? No. Okay, you can have microorganisms growing within your tissues, dropping some nutrients, but they're not harming the host in any noticeable way. If that's the case, we're said to have an inapparent, or sometimes the word subclinical infection occurs. So let's make sure we're on the same page. Infection means you've got microorganisms growing in your tissues. Tissues are normally sterile in terms of microbial growth. So something's growing there that shouldn't be there. But if the teeter-totter of balance between the parasite obtaining nutrients and damage to the tissues as a result of that is pretty balanced, then you can just harbor these organisms and they're apparent in this, they're present in this inapparent or subclinical state. And we don't end up having any disease symptoms. And that's kind of an ideal situation for the parasite. Obtain some food, but don't overly harm your host. What happens if that infection gets out of control and the teeter-totter shifts balance to damage? Okay? And now the growth of these microorganisms is causing appreciable damage to the tissues and the organisms. Then we switch over and have a clinical infection. We have an obvious disease state with some more than likely noticeable symptoms associated with that disease state. Well, now we've got a full-blown clinical infection. And now the parasite's in some jeopardy because it could kill its host. And if it kills its host, what does it have to do? It's got to find a new host, right? And that isn't always easy. It's not always easy for a parasite to jump from one host to another. You know what we call diseases that involve a parasite moving from one host to another? They're called communicable diseases. Okay? If a parasite kills its host, it's oftentimes referred to as an inept parasite. Not all parasites are created equal. Some are potentially more damaging than others. Some pathogens are more potentially deadly than others. And so a term that you need to know is the term virulence. Virulence refers to the ability to cause a disease state. Some pathogens are highly virulent, others not so much. Depends upon the nature of the pathogen and what it's doing to cause that particular disease state. And that's something we'll talk about a fair amount later on as we get into this topic, because for particular diseases that are caused by microorganisms, how is that pathogen making you sick? What is it doing? What are its characteristics that cause it to be a pathogen, and either a highly virulent pathogen or maybe a fairly lowly virulent pathogen? A concept that's related to virulence is the concept of infectious dose. Infectious dose refers to how many cells of a pathogen do you need to cause disease. So what's going to be the relationship between infectious dose and virulence? Typically, highly virulent pathogens can have a very low infectious dose. It doesn't take many cells to cause a disease state. Pathogens that have a low state of virulence, you need a whole bunch of cells to make you sick. And so there tends to be an inverse relationship between infectious dose and virulence. Got a little bit out of order here. It's okay. Okay. This relationship between pathogenic microorganisms or non-pathogenic microorganisms also all begins the same way. The microorganism has to make contact with the host in order for this relationship to begin. And an important part about contact is microorganisms have to be able to adhere to the host to be able to gain entry into the tissues. And we're going to get into that in a little bit more detail in a minute. But all these relationships that we talked about, commensalistic relationships, mutualistic, parasitic relationships particularly, begin with the phenomena of contact. Okay? As a result of contact, you can become permanently colonized with a group of microorganisms that stay with you much of your life. You can acquire that normal flora. And as I said to Arthur, as the baby moves through the birth canal, it begins to acquire a normal flora during that process. One of the things that's of interest to researchers is, what is the difference in the nature of the normal flora that develops between vaginally born babies and C-section babies? Because they have a different route into the world. And that influences the normal flora that they acquire. If the microorganisms invade the tissues, then we have a state of infection. So those microorganisms are multiplying within the tissues. It's very likely that your defenses will hold these microorganisms in check, and that there won't be any real disease state associated with the infection that you're harboring. So we'll have that subclinical or inapparent infection. And eventually, the immune system probably is going to deal with those invaders and eliminate them. And hopefully any damage that they've done, even the small damage, will be resolved. And so you will recover, probably never even knowing that you had these things growing within you. But if the microorganisms get the upper hand, then they can begin damaging the tissues, causing a state of disease. And if that damage to those tissues is severe enough, you can die. Mortality. The term morbidity refers to the incidence of disease within a population, how many cases are occurring. You're going to see these terms, morbidity and mortality, in another context later on when we talk about epidemiology. It's also possible that you may maintain a more or less permanent carrier state where microorganisms are established in your tissues, but the disease isn't apparent. In this particular case, you are said to be a carrier. And you can transmit that disease to another individual even though you're not showing symptoms yourself. One of the most notorious carriers was in I think it was the 1920s, maybe 1930s. A woman by the name of Mary who harbored typhoid bacillus in her liver. And she was constantly shedding those bacillus into body excretions, and she worked in the food industry. This was the infamous Typhoid Mary, who caused untold number of cases of typhoid and the outbreak of typhoid epidemics when she was working in the food industry and unintentionally exposing people. She's a very interesting story. Once they discovered what her status was, she was basically banned from working in food industry, and she didn't really have any other skills, and so she kind of defied that ban and moved to other cities and had false names and things like that, and caused a lot of outbreaks all around the country. And so the story of Typhoid Mary is a very interesting story. It's kind of a sad story. This normal flora that we all have and acquire throughout our lifetime is really important to us. These are some of the sites of the body that harbor a normal resident flora, and these are kind of the parts of the body that the Human Microbiome Project is trying to catalog and determine what is the typical population of microorganisms that exist in each of these particular areas. We did just a little minor overview of your skin and mucous membranes to try to get a little bit of a handle on just a couple of species that were your normal flora. I think, wasn't it Taylor that did a swab of her eye? Yeah, she got some kind of nasty looking stuff there. Some interesting research has been done to try to kind of document the importance of the normal flora to the functioning of us as humans. And one of the ways you can do that, this is just an electron micrograph of some human tissue with some normal flora bacteria associated with it. One of the interesting branches of research that deals with the effect of normal flora on your body is to grow animals in a germ-free state. They deliver them in sterile operating rooms by cesarean section, and then they immediately transport them into sterile environments where they're not exposed to any microorganisms. So these particular animals are called azenic animals. They're germ-free animals. And you might think being germ-free would be beneficial because you wouldn't get sick as much. But as it turns out, there are some interesting consequences of not having a normal flora of being a germ-free animal. We tend to get an enlargement of the cecum, and other degenerative diseases of the intestinal tract tend to be much more pronounced in germ-free diseases. What's the significance? We think that these normal flora microorganisms are necessary for normal intestinal development. This is a whole interesting area of medicine. There are a whole lot of bowel inflammation diseases. You might have heard of Crohn's disease before, or irritable bowel syndrome. There are all kinds of bowel inflammation diseases, which are really nasty and really of major consequence health-wise to a number of people in this country. And there's now a lot of research that's gone into the role that the normal flora and the type of microorganisms you carry in your bowel plays in relationship to developing this particular disease or set of diseases. There's even some therapies that are associated with trying to reestablish your normal flora if you suffer from some of these diseases. And so they have done, for example, fecal transplants, where fecal material from a person with a normal bowel is transplanted into a person suffering from one of these inflammatory bowel diseases. And they've had some pretty good success with those kind of transplants. They've had lyophilized and dehydrated normal flora bacteria that are placed in pills that people will eat. They're affectionately called poop pills. And they're used as a way to try to reestablish a normal flora, to try to correct these inflammatory diseases, which are so debilitating. Germ-free animals tend to have vitamin deficiencies many times. That's because the normal flora bacteria oftentimes produce vitamins and secrete them into the gut. This is a huge one. Underdevelopment of the immune system in most animals. The normal flora that we acquire is really, really important in priming or kind of setting off your general development of immunity. That might not mean to you much right now, but later on you're going to learn that in the immune system, in order to develop immunity, you've got to be challenged by foreign substances called antigens. And these normal flora contain antigens which can challenge and prime your immune system and get it ready to go without causing disease. So they're really important. Lessen sensitivity to various other types of diseases, including enteric pathogens, some type of amoebic dysentery. Some of these normal flora bacteria actually are aiding these pathogens. And so that wouldn't be an antagonistic relationship. That would be a synergistic relationship. So in this case, the normal flora are causing some damage in some particular situations. Less body fat. We think that the normal flora help to break down and digest things that your body might not normally digest as well. So being born in this particular germ-free state has some very negative consequences. This is a chart that is just here for illustrative purposes. It's not anything that I would want you to even begin to try to memorize or anything like that. But this shows you some of the well-established microbiota that's been identified in various parts of the body. Okay, so for the skin, for example, that's the only one we'll look at right now. Okay, bacteria, what do we find? We find a lot of Staphylococcus, some Micrococcus, some Streptococcus, for example. And so that's one of the reasons why we are concentrating on Staph and Strep in terms of trying to identify some representative species of your normal flora. But a very different group of organisms inhabiting the gastrointestinal tract, for example. You see different species names here from the bacteria. Large intestine tend to be different from the oral tract. Upper respiratory tract, different microorganisms. And so each one of these parts of the body, which is a unique little microhabitat, tend to have different resident microorganisms. Generally speaking, unless there's a disease state, the internal organs and fluids of the body should be germ-free. And that's one of the criteria that we use to try to establish whether infection has occurred, is to sample tissues and look for the presence of pathogens, particularly in the fluids, the blood and the cerebrospinal fluid. Okay, anybody ever seen the spinal tap done before? Okay. One of the things with the spinal tap is the cerebrospinal fluid should be clear. And if you have bacterial infection in that, like meningitis, then the cerebrospinal fluid is cloudy. It's turbid, much like your broth tubes are turbid when there's growth in it. Blood should be pathogen-free. If you've got pathogens in your blood, then you've got a case of septicemia. And septicemia is really deadly. That's one of the reasons why I was so surprised that the blood that we got, the sheep blood we got, had contaminants in it. We called the company that sold it to us and said, hey, you should be aware of this because there should not be pathogens in that blood. That blood should be sterile in terms of not having infectious agents. The heart, the liver, the lungs, bones, all of these areas should be micro-free. And if they're not, then that typically indicates a state of disease. Okay, some of these terms I've used already, others I haven't. I'm going to ask you to take a look at this particular slide so that you can begin to become familiar with the terminology associated with disease. I talked a little bit about asymptomatic, which is the same thing as subclinical or inapparent disease. Mentioned communicable and non-communicable. But there are some other types of terminology that I don't want to read to you, but I would like you to look through here and start getting a handle on what these particular terms refer to. I talked about the concept of virulence and the relationship between virulence and infectious dose. This shows you estimated infectious dose of a particular pathogen. So compare the infectious dose estimate of measles with that of cholera. Which disease is more virulent? Measles is highly virulent. cholera, very lowly virulent. Look at the number of cells that you've got to get, that you've got to ingest through probably drinking contaminated water to get the disease cholera. So one of the things we're going to talk about is, why do some organisms have such a low infectious dose? Why are they so virulent? And why are other organisms so weakly virulent? Why do they have such a high infectious dose? Typically what we're talking about is different organisms have different adaptations that allow them to penetrate the tissues, evade the immune system, and begin to establish that particular disease state in your body. So we're going to be talking about some of these things again, some of these infectious agents, and specifically what adaptations do they have that make them virulent pathogens. We said that in order for disease to develop, it had to begin with contact. Contact involves then adherence, where the organism has got to kind of latch on. When we talked about bacterial structures at the beginning of the semester, we talked about some bacterial structures that allowed pathogens to adhere to the surface of tissues or to other cells. Things like fimbriae, for example, capsules, in some cases the glycocalyx. Generally speaking, if the pathogens can become involved in a biofilm, then they have a much higher chance of staying adhered to the tissues and then gaining penetration into the deeper underlying tissues. But typically, in order for infection to occur, a pathogen has to have some defined portal of entry. It's got to have some way to get in. Different pathogens have different portals of entry. For some, it is inhalation. For some, it is ingestion. For some, it might be injection, broken skin, an insect bite. The pathogen might be transmitted sexually. The conjunctiva of the eye is a very, very common portal of entry. If you think about it, your eyes are pretty defenseless. They're constantly being bombarded with dust particles, and those dust particles can contain pathogenic microorganisms. Your eyes do have a couple of adaptations, however, to try to prevent infection. One of them is the secretion of tears, which kind of washes the dust off your eyes. And in those tears, there's the enzyme lysozyme, which tends to break down bacterial cell walls and cause cell lysis. Many times, these portals of entry are associated with portions of the body that already have a well-established microbial flora. Another term I didn't give you, but I should. Your microbial flora refers to kind of your more or less permanent group of microorganisms that inhabit your body. Now, I did say earlier that your flora can change over time. Your flora develops as you age. Part of that is hormonal, so hormones can have an effect on normal flora. Drug treatment can have an effect on your flora. Antibiotics particularly can do a nasty job of wiping out your intestinal normal flora. That's one of the reasons why doctors now are oftentimes recommending that you take probiotics or eat yogurt, for example, with active microbial cultures in it. If you're undergoing antibiotic therapy, try to reestablish or reseed your normal flora. I mentioned age. Age, hormones, diet can affect your normal flora. And so it's not a fixed thing. But the normal flora is different from the transient flora. Transient flora are things you pick up, but you don't keep very long. Why? Because typically your normal flora is going to eliminate the transient flora. It's going to outcompete it. So one of the things that those of you that tested positive for Staph aureus on your skin might be interested in doing is doing that test again by the end of the semester and see if you still test positive for Staph aureus. Because it would be interesting for you to know whether you are a kind of a permanent carrier of that, or whether that's something you just picked up and then you've gotten rid of. Transient flora can cause disease. But typically, that only happens when you have a lowered state of immunity. What's causing that lowered state of immunity? You're sick. You have some type of a primary infection. And that's putting some stress on your immune system. If your transient flora, something like Streptococcus pneumoniae, causes disease because of your lowered state of immunity because of the primary infection, then this particular disease is called a secondary infection. Pneumonias are notorious secondary infections. People get sick with something, their immunity is lowered as a result of that, and then transient microorganisms that normally wouldn't be harmful cause a disease in that lowered state of immunity. That's one of the reasons why they recommend that older people get pneumonia shots, for example, that they make sure that they get their flu shots, for example, to try to prevent these secondary diseases from gaining a foothold just because your immune system is normally lower. Well, if the portal of entry is breached, then the microorganism that causes disease, if it becomes stuck onto the tissues, if adhesion occurs, can invade those tissues, multiply. They can infect some target tissue and damage that tissue and result in a disease state. Oftentimes, the pathogen is then shed from the body so other people can be infected at some type of a portal of exit. And the portal of entry and the portal of exit are usually different. What's really tricky for a diagnostic physician is to be able to look at someone who is sick and has some definable symptoms as a result of their illness, and being able to correlate those particular symptoms with a particular pathogen or group of pathogens. Because you need to know what someone has in order to effectively treat them. And so that whole art of physical diagnosis, of taking a history, observing a person's symptoms, and then being able to diagnose what's causing that, is really an art and a very important part of the practice of medicine. And I think those of you that did the JMH rotations hopefully saw how important it was to take good histories in terms of the ability to effectively diagnose and treat disease. This is just showing some of the adaptations that microorganisms can have that allow them to attach to tissues and then penetrate and multiply and cause infection. Things like fimbriae, capsules. On virus, there are various envelope proteins oftentimes, or capsid proteins that are receptors for the surface of a host cell. But if you don't attach, then you're typically not going to cause disease. And so that process of attachment is really important. This shows you the relationship between receptors on the surface of a microorganism and receptors on the surface of an infected host cell, almost kind of like docking sites that allow that particular microorganism to attach to the cell surface and probably eventually be brought in to that cell by endocytosis. Okay, so next time we're going to talk about virulence factors. And what are virulence factors and why are they important? So tomorrow we'll set up some dilution plates so you can count some microorganisms and figure out concentrations of number of cells in unknown samples. Yeah, I'll have the grades. I've got to have the grades turned in by noon, and I've got a meeting right afterwards. So I might get them into the lab in your drawers this afternoon. If I do, I'll send you an email this. But definitely have them tomorrow morning if you don't have them this afternoon.